The definition of a closed-system drug transfer device from NIOSH stipulates that the device mechanically prevent the transfer of environmental contaminants into a system and also prevent the escape of the hazardous drug or vapor outside of the system. Recently, the University of Utah completed a scientific study to evaluate vial transfer devices for containment of hazardous drug vapors. This study has become known as the Utah Protocol. There are numerous systems available for medication preparation, and many systems market themselves as closed. The purpose of this study was to examine several available systems to determine which drug transfer devices prevent the escape of vapor outside of their system. Titanium tetrachloride was selected for this study to simulate the escape of vapor from a CSDTD. Titanium tetrachloride generates very visible smoke when it comes into contact with moisture in the air and is a standard tool in air visualization studies. Simulations were conducted in a research laboratory at the University of Utah Moran Eye Center using a Niederman extraction arm mounted to the Moran Eye Center ventilation system to remove any of the potentially irritating smoke. 3 milliliters of titanium tetrachloride was delivered into a glass vial and sealed with a 20 milliliter crimp seal containing a PTFE membrane and silicone backing. Sealing was accomplished with a firm press H2O7 aluminum crimper. The transfer devices were assembled and each device spiked its own vial containing titanium tetrachloride. A 60 milliliter Lure-Lock syringe from Becton Dickinson & Company was filled with 50 milliliters of air and attached to each device. The transfer device with the attached syringe was placed under the Niederman extraction arm for photography, videography, and ventilation. The plunger of the syringe was compressed at a constant rate over 5 seconds. Photographs and a video were taken to document any escape of smoke from the transfer device. Five different products were tested in the Utah Protocol. The first product tested was the Tevadaptor Syringe Adapter System with the Tevadaptor Vial Adapter System by Teva Medical Limited. The Tevadaptor has a 0.2 micron air filter and a carbon-based filter. The syringe adapter allows for the attachment of a standard Lure-Lock syringe. The Tevadaptor was spiked into the vial of titanium tetrachloride and connected to the syringe adapter to create the transfer system. A second product tested was the Facile Protector 50 and the Facile Injector Lure-Lock by Carmel Pharma. The Facile Protector 50 is a mechanically closed pressure equalization device attached to the vial to prevent a vacuum or excess pressure with the preparation of drugs. The Facile Injector Lure-Lock is attached to the Protector 50 to ensure closed system transfer of drugs. The protector was attached to the titanium vial utilizing a Facile M12 assembly fixture. A third product tested was the Chemo Mini Spike Dispensing Pin by B. Braun Medical Incorporated. This device has a 0.2 micron air filter and a 5 micron fluid filter and connects to a normal Lure-Lock syringe for the preparation of cytotoxic agents. The dispensing pin was spiked directly into the vial of titanium tetrachloride and connected directly to the 60 ml BD syringe. A fourth product tested was the Alaris Smart Sight by Cardinal Health. The Smart Sight has a 0.2 micron hydrophobic air venting filter and allows for needle-free access to the vial via a normal Lure-Lock syringe. The Smart Sight was spiked directly into the vial of titanium tetrachloride and connected directly to the 60 ml BD syringe. The last product tested was the ChemoProtect Spike by Codan. The ChemoProtect Spike has a 0.2 micron air filter and a 5 micron liquid filter and connects to a normal Lure-Lock syringe. It was spiked directly into the vial of titanium tetrachloride and connected to the 60 ml BD syringe. Only the Facile system prevented release of smoke in all tests. All of the filter-based systems allowed smoke to move from the vial into the work area in all tests. The Facile system by Carmel Pharma was the only system tested that completely prevented the release of smoke and meets the definition of a closed system transfer device as stated in the NIOSH alert. Systems using traditional 0.22 micron filters do not contain the release of gas. If carbon-based filters are utilized as in the Teva system to trap the active drug component present in gas, the actual performance in this regard must be demonstrated in published peer-reviewed journals as stated by the ASHP guidelines. To validate the test, an independent testing laboratory was provided with the testing materials and protocol, and the test was duplicated. Results from the independent laboratory were identical to the results seen at the University of Utah, with only the facile system being closed. Also, to eliminate any concerns regarding filter damage from the hydrochloric gas which would potentially contribute to the escape of the smoke from the filter-based systems, the same independent testing laboratory was asked to verify filter integrity. Following the test, filters were removed and coated with gold and dried and viewed under high-powered electron microscopy. There were no breaches in filter integrity from the brief exposure to the hydrochloric gas that could have contributed to the observed escape of smoke.